Hi, my name is Taylor Reiter. I am a postdoc in the Green Lab at the University of Colorado Anschutz, but today I'm going to be telling you about a tool that I helped develop during my PhD with Titus Brown in the DIB lab at the University of California, Davis. The tool is called Charcoal, and it helps detect and remove contamination in genomes. So public databases are rife with contamination, and I think that this is a fairly well appreciated problem at this point. Here I'm showing a plot from a recent publication, Contaminator, and it shows us that both in RefSeq and in GenBank, across the different domains of life, there can be a lot of contamination. And I think these estimates really vary based on which tool is used, but we can see that they span from anywhere between like 5% of genomes to up to 90% of 5% of species and 90% of species, depending on which database and which kingdom that we're looking at. So given that this is such a problem, the contamination detection tool space is actually quite robust. But a lot of these tools, they sort of have two problems. The first is that most of the tools don't actually remove the contamination. So for microbes, a really popular tool to use is one called CheckM. And it gives pretty good estimates about the amount of contamination in your genome based on marker genes, single copy marker genes for the lineage of your genome. But once you get an estimate of the contamination, that contamination is not removed. You have to use a different tool that's much more computationally intensive to go back and detect or to go back and remove that contamination. And then similarly, precise methods don't scale to all of the genome. So we have some tools that use like long K-mer matching, but Contaminator, for example, uses all versus all alignment, and that doesn't scale very well. It needs to be redone every time that you have additional genomes if you would like to detect contamination. On the flip side, long K-mers are very accurate for giving you species level designation or even sometimes strain level of what genome you're dealing with. And so here I'm showing how different tools performed when trying to figure out what species were in a metagenome, which can kind of be is a similar problem to detecting contamination in a genome because you end up with multiple species like you would have in a metagenome. So these are different metrics where completeness is the ratio of taxa that are correctly identified in the ground truth, or purity, the ratio of correctly predicted over all predicted. And we can see that SourMash, a tool that uses long K-mers to match against a database, performs really well, especially compared to other tools. So the exact command that SourMash uses to do this is called SourMash gather. So taking advantage of this property of SourMash and property of really long K-mers, we developed a tool called Charcoal. So Charcoal applies long K-mer matching against databases to detect and then remove contamination. And this tool is powered by SourMash gather and then a lot of glue to hold all of the code together and remove that contamination. So the outline for the rest of this presentation, I'm going to give an overview of the charcoal decontamination workflow. I'll then show you some charcoal results on simulated genomes where we know the answer of whether something's a contaminant or not. And then I'll show you results for applying charcoal to all of the GTDB genomes. And lastly, show you some impacts of how using a different database changes your charcoal results. So starting off with the charcoal decontamination workflow. What Charcoal does is it creates a FracMin hash sketch, so a min hash sketch for each of the contiguous sequences or scaffolds that are in your genome. It then compares that sketch against a database and returns all of the genomes that contain K-mers that are in those contiguous sequences. Using that information, it determines the taxonomic lineage of each of your contiguous sequences. Then it votes and it says, okay, most of your contigs have this lineage up to whatever user specified level. Usually the order works pretty well. And it says all of these contigs agree at this order level, but this one doesn't. So that is a contaminant contig. And then it produces a summary based on those results. After this initial contamination detection stage, you can do one of three or all three of these downstream things. So the first is you can verify the contamination. So taking the contiguous sequence that was a contaminant, it will download the genomes that match to that contig, map them against, map the two contigs together, and then show you what that alignment looks like. There's also some visualizations and reports that can be generated. And then lastly, you can tell Charcoal to clean and remove the contiguous sequences. It splits them into a clean and dirty file, so you still have access to the sequence, but they're split apart, so you can see charcoal's determination there. So how does charcoal perform on simulated genomes? Charcoal is actually fairly conservative as a decontamination approach. So here I'm showing some simulated genomes that were created by the tool Gunk, and each genome had 3 genomes that either varied at the phylum, class, order, family, genus, or species level. 1/3 was the correct genome, so we told charcoal this is the actual lineage that is in our genome, and then the other 2 were contaminants. And we can see that in this light green, our actual genome is very rarely demarcated as contamination, so that's good. We don't throw out any real genome sequence. But then in blue, we see the contamination. So dark blue means charcoal detected it as contamination, and light blue means it was contamination and it wasn't thrown out. So we only see dark blue at the phylum, class, and order level because charcoal by default only decontaminates up to the order level. So nothing really gets thrown out at the species, genus, or family level. But then we do have a lot of light blue sort of left over. So we were curious, why is charcoal missing some of these contigs? And the reason why is because in these simulated genomes, a lot of these contiguous sequences are very short. So if we look at the average length of contig, or really just the length of contig across these different taxonomic levels, a contaminant sequence is much less likely to be called a contaminant by charcoal when the contig is much shorter. So on average, about 2,700 or 3,700 base pairs versus about 6,000, 7,000 base pairs. So charcoal is much less reliable for very short contigs, which we would expect because you need to have many k-mers in order to match correctly against the database, and short contigs have fewer k-mers on them. Okay, so what happens when we apply charcoal to the GTDB genomes? Charcoal removes contamination from about 25% of the genomes in GTDB. This contamination is really distributed across all of the genomes. So it's in both the GenBank, RefSeq, representative, and not representative, although the representatives are less likely to have contamination, which is good. It occurs across basically all of the orders that are in the GTDB. And then NCBI genome categories, we thought that maybe metagenomes or environmental samples might be more likely to have contaminants, but genomes that come from single cell, environmental, or isolate, they're all contaminated. Or we observe some contamination in all of them. And again, like with the simulated genomes, contamination is less likely in genomes with longer contigs. Okay, so database, how does that impact our charcoal results? Because charcoal is database dependent. We're comparing our k-mers and our contiguous sequences against those in a database. So larger databases discover more contamination when we use our simulated genomes. So here I'm showing you an upset plot for the phylum level genomes, and 3,600 contigs are detected as contaminants by all of the different databases. GTDB full or REPS. So REPS is a subset of the full and has about 60,000 genomes in it, and the full has just over 300,000. But 1,000 contigs are only detected as contamination when we use the full database. And what's happening here is that a lot of the contiguous sequences, while they're identified by both databases, many of them are actually only ID'd when you use the full database and not the REPS database. And that's because you have many more k-mers matched against the full than you have against the REPS. And charcoal requires that you have at least 3 contigs match. And so you have more opportunities to match against the full database than you do against the REPS. So that's why we recover more. But something interesting happens when we use full GTDB. So larger databases discover less contamination in real genomes. And so we can see here, these are this is a subset of GTDB, so it's about 1/20th of RS202 or 1/25th of RS207. And we get good agreement in about 1,200 genomes across 3 of our databases. But then the REPS actually discover more contamination in 2,000. And we're still digging into this result, but we think what's happening is that because the reps themselves are contaminated, it shields some contamination from discovery. So more on that later. And that is the charcoal tool. So thank you for your attention, and I welcome any questions at my email address here.